this video, we're going to talk about proteins, enzymes, and the chemistry of life. Here are your learning targets. Every single day, your body comes into contact with millions of microbes, many of which have the potential to cause disease. You breathe in shared air, touch contaminated surfaces, and eat food that isn't sterile. Yet, most of the time, you don't get sick. Why? Because deep inside your body, a specialized defense system is always working fast, quiet, and incredibly precise. At the heart of that system are called antibodies, your body's natural surveillance and response team. Antibodies, also known as immunoglobulins, are Y-shaped proteins produced by B cells, which are a type of white blood cell. They belong to your adaptive immune system. This is your body's smart defense team. So it fights off specific germs like viruses or bacteria and remembers them. So if they come back, your body can respond faster and stronger. It's like when you get infected with chickenpox virus for the first time. Your immune system remembers it. It will be stopped quickly when you get infected again. So each antibody is designed to bind to one specific antigen. Again, an antigen is a molecule found on things like viruses, bacteria, or even the wrong blood type. Once an antibody locks onto its target, it either blocks the threat directly or tags it so other immune cells can destroy it. Now let's break down the Y shape. Each antibody is made up of 4 protein chains, 2 heavy chains, and 2 light chains on the outside. So these chains are held together by disulfide bonds to form the classic Y shape. At the tips of the Y is the variable region. This part is different in every antibody, allowing it to recognize and bind to a specific antigen. Think of it like a custom-designed key for a specific lock. Then you also have the constant region, which provides structure and communicates with the immune system. Each of the Y arm is called a Fab region. It includes both the variable and part of the constant region. This is an antigen-grabbing part that attaches to invaders and holds them in place. This is called Fab because it's fragment antibody. It's the one that connects to the antigen. The stem of the Y is the Fc region, or fragment crystallizable. It doesn't grab the enemy, but it sends signals to the rest of the immune system, calling in other cells to attack or clean up the threat. And of course, the disulfide bonds. They hold the chains together and keep the Y shape stable. Basically, the Y shape allows antibodies to grab specific invaders or antigens at the tips and call for the help at the base. Here's the table comparing the different parts of the antibodies. In the previous video, you've learned how antibodies help identify blood types by attacking red blood cells with antigens you don't have, causing clumping if mismatched. But antibodies do much more than that. They are the immune system's targeted weapons, made to stick to specific invaders like viruses or bacteria. Once they bind, antibodies can stop the threat, tag it for destruction, or block it from spreading. They also leave behind immunological memory, so next time that the same pathogen shows up, your body reacts faster. That's how vaccines protect you. Outside the body, scientists use antibodies in diagnostic tests, like COVID kits and pregnancy tests. This includes cancer therapy and autoimmune disease treatments. However, when antibodies go rogue and attack your own healthy cells, we call that autoimmunity. Understanding antibodies help us treat and even prevent these conditions. Now that we've talked about how your body defends itself, let's look at how it sustains life every second through oxygen transport. Hemoglobin is a large folded protein found inside your red blood cell. It's responsible for carrying oxygen from your lungs to every cell in your body. This molecule is made up of 4 protein chains, 2 alpha and 2 beta. Now each chain is attached to a heme group, which contains an iron ion at its center. So that iron is key, because it binds directly to oxygen. Since there are 4 heme groups per hemoglobin molecule, each one can carry up to 4 oxygen molecules at once. This process is reversible. Hemoglobin picks up oxygen where it's abundant, like in the lungs, and releases it in places that need it most, like active muscles and organs. Now, hemoglobin isn't just an oxygen taxi. It's smart about when to hold on and when to let go. In the lungs, oxygen is everywhere. Hemoglobin has a high affinity here, meaning it loves oxygen and grabs it easily. But in the tissues, especially muscles that are working hard, oxygen levels are low. Hemoglobin's affinity drops, so it starts to release oxygen right where the body needs it. Now, this release is controlled by something what we call Bohr effect. When tissues are active, they become more acidic, produce more carbon dioxide, and have lower oxygen pressure. These changes tell hemoglobin to let go of that oxygen. And there's more. Hemoglobin also shows cooperative binding. When one oxygen molecule binds, it changes the shape of the hemoglobin, making it easier for the next oxygen molecules to attach until all four are bound. All of this makes hemoglobin a beautifully regulated, responsive protein. It is perfectly designed to meet the oxygen needs of every part of the body. This slide shows the summary of conditions that affect hemoglobin's oxygen binding. In the lungs, there's a lot of oxygen, so hemoglobin grabs it. So that's high affinity. In the muscles, where there's low oxygen, more carbon dioxide, higher temperature, and more acidity, hemoglobin lets oxygen go. Now let's explore 3 disorders that affect hemoglobin. These conditions may differ in cause, but all interfere with how oxygen reaches the tissues. Number 1 is sickle cell anemia. This is caused by a mutation in the beta globin gene, where valine replaces glutamic acid. So this forms hemoglobin S or HbS, which tends to clump together under low oxygen. This causes red blood cells to become sickle-shaped, making them stiff and prone to blockage in blood vessels. These cells also break down easily, leading to anemia, pain crisis, and organ damage. Next is thalassemia. This condition is due to reduced or absent production of alpha or beta globin chains, resulting in imbalanced hemoglobin synthesis. Red blood cells become small, pale, and fragile. This causes chronic anemia. Patients often require frequent blood transfusion, which may lead to iron overload. Now, it is most common in the Mediterranean, Middle East, and Southeast Asia, where carriers had a survival advantage against malaria. But inheriting 2 defective genes leads to the disease. Next is methemoglobinemia. Here, the iron in heme is oxidized from Fe2 plus to Fe3 plus, forming methemoglobin, which cannot bind oxygen. As a result, oxygen delivery is impaired, even with normal number of red blood cells. Symptoms include cyanosis or bluish skin and tissue hypoxia. So this condition may be inherited or triggered by drugs or chemicals, such as nitrates and certain anesthetics. These conditions highlight how changes in hemoglobin structure, production, or chemistry can drastically impact oxygen transport and overall health. So far, we've seen how proteins like antibodies and hemoglobin help protect and transport. But there's another class of proteins that control how fast your body works, and these are enzymes. Enzymes are special proteins that act like biological catalysts. They speed up chemical reactions in the body without being used up or permanently changed in the process. Now, each enzyme is specific. It only works on one type of molecule called its substrate. The substrate fits into a special region of the enzyme called the active site. It's like a key fitting into a lock. They're very specific. The active site is where the reaction happens. And here's the cool part. Enzymes work by lowering activation energy. When we say activation energy, that's the minimum energy needed to start a chemical reaction. Because of this, reactions that would take hours or never happen at all can occur in just seconds. And once the job is done, the enzyme is ready to do it again. Now, let's talk about how enzymes interact with their substrates or molecules they work on. First, we have the lock and key model. In this model, the enzyme's active site has a fixed shape that fits only one specific substrate. It's like a key fitting into a lock. The idea here is if the shape doesn't match, the substrates won't bind. This helps explain why enzymes are highly specific, but it has a limitation. It assumes the enzyme never changes shape, which we now know isn't always true. That's where the induced fit model comes in. It builds on the previous model by saying that the enzyme is actually flexible. When the substrate approaches, the enzyme slightly adjusts its shape to fit around it, kind of like a hand forming around a stress ball. This makes more sense with what we've observed in real enzymes. Many enzymes show this flexible behavior, which helps them recognize a range of similar substrates, not just one perfect fit. So to summarize, the lock and key model is simple and explains specificity, but in the induced fit model, it better explains the actual dynamic interaction between enzymes and substrates. Now, enzymes are powerful, but they cannot run wild. If they're always active, the body could waste energy or make too much of something, even harmful substances. That's why enzyme activity must be carefully regulated. So first, let's talk about competitive inhibition. Here, a molecule that mimics the real substrates sneaks into the active site. Because of that, the real substrate cannot bind. Therefore, the reaction slows down. Next is non-competitive inhibition, also called as allosteric inhibition. This time, the inhibitor binds to a different part of the enzyme, not the active site, rather the allosteric site. But this still causes trouble by changing the enzyme's shape, so the substrate no longer fits. So the result, even if the substrate is around, the enzyme stops working well, and the reaction rate drops. Finally, we have the feedback inhibition, or the end product inhibition. This happens in long reaction chains called metabolic pathways. When there's already enough of the final product, it goes back and shuts off the first enzyme in the sequence. It usually binds to an allosteric site to change the enzyme shape, like a stop signal. If product levels are high, production slows down. When levels drop, the enzyme gets reactivated and the process restarts. So these three inhibition types, they help the cells save resources, avoid overload, and stay in balance. As mentioned, enzymes are very powerful, so we can also actually use them for diagnosing diseases. Here, we look at two key enzymes, ALT and AST. ALT, or alanine transaminase, is mostly found in the liver and helps break down amino acids. AST, or aspartate transaminase, also helps with amino acid metabolism, but is found in several organs, like the liver, heart, and muscles. When these enzymes are elevated in the blood, it usually means liver cells are damaged, maybe due to hepatitis, alcohol, or certain drugs. If AST is higher than ALT, it may also point to muscle injury. Just remember, ALT almost liver targeted, AST affects several tissues. Next, we have heart and muscle markers. Troponins T and I are not enzymes, but they're crucial markers for heart damage. They help regulate heart muscle contraction and rise quickly after a heart attack. CKMB, or creatine kinase muscle brain time, is an enzyme that gives energy to muscle and heart cells. High levels usually mean there's been muscle damage or a heart attack. Now for pancreas, amylase helps break down starch, while lipase breaks down fats. When the pancreas is inflamed, like in pancreatitis, these enzymes leak into blood, so doctors check their levels to confirm for the condition. And finally, for blood glucose monitoring, we use an enzyme called glucose oxidase. This enzyme reacts specifically with glucose to produce a tiny electrical signal. That's how glucose meters give you your blood sugar reading. This test doesn't measure the enzyme itself, but it uses it to detect glucose accurately and quickly. Basically, these enzymes act like molecular clues. When they are too high, or when they show up where they shouldn't, they help doctors pinpoint what's going wrong in the body. Now, some of the most common medicines we take actually work by blocking enzymes. Let's look at 4 examples. Statins or statins are used for high cholesterol. They block the liver enzyme that makes cholesterol, so your body makes less of it. ACE inhibitors are for high blood pressure. They block the enzyme that tightens your blood vessels, helping them relax and lowering your blood pressure. ACE stands for angiotensin converting enzyme. Protease inhibitors are used in HIV treatment. They block the viral enzyme AHB needs to make new virus parts. This slows down the infection. Lastly, aspirin is for pain, fever, and swelling. It blocks the enzyme that makes pain and inflammation chemicals, so you feel better. The key idea here is that enzymes are specific. So if a drug can target just one, it controls specific body functions, like lowering cholesterol or stopping pain, without affecting everything else. So far, we've talked about how enzymes work and how the body controls them. But what happens if your body can't make certain enzymes at all, or if the enzyme doesn't work the way it should? Some people are born with genetic mutations that affect enzyme function. These are called inborn errors of metabolism. When an enzyme is missing or defective, certain substances can build up to toxic levels, or the body may not make essential products it needs to survive. Two important examples that we'll study are phenylketonuria, PKU, and G6PD deficiency. These can be life-threatening if left untreated, but manageable if caught early. Phenylketonuria or PKU is a genetic disorder that affects how the body processes phenylalanine. It is an amino acid found in protein-rich foods. In people with PKU, a mutation in the gene for the enzyme phenylalanine hydroxylase, PAH, means that phenylalanine isn't broken down properly. Now let's walk through what happens in phenylketonuria. It starts when you eat protein-rich foods like meat, eggs, or dairy. These foods contain an amino acid called phenylalanine. Normally, the body uses an enzyme called phenylalanine hydroxylase to convert phenylalanine into another useful amino acid, tyrosine. Tyrosine is important because it helps make brain chemicals and pigments the body needs. But in people with PKU, the PAH enzyme is missing or defective because of the genetic mutation. So phenylalanine doesn't get converted. Instead, phenylalanine builds up in the blood, and at high levels, it becomes toxic to the brain. If PKU isn't treated early, this buildup can lead to intellectual disability and other serious symptoms. That's why newborns are screened for PKU, so they can start a special low phenylalanine diet right away, and of course, prevent long-term damage. Next, we have G6PD deficiency, or glucose 6-phosphate dehydrogenase deficiency. G6PD deficiency is a common inherited condition where the body doesn't make enough of the enzyme G6PD. This enzyme helps protect red blood cells from substances that causes oxidative stress. Oxidative stress is a type of chemical change. It starts with a mutation in the G6PD gene. This affects how much of the enzyme glucose 6-phosphate dehydrogenase the body makes. As a result, the number of red blood cells have less G6PD enzyme activity. This enzyme is important because it helps red blood cells produce NADPH. It is a molecule that protects them from oxidative stress. Without enough NADPH, the red blood cells lose their defense against reactive oxygen species, or ROS, damaging molecules that build up in the body. Now, this oxidative stress can increase during infections, after eating certain foods like beans, or when taking specific medications like sulfa drugs. When reactive oxygen species, or ROS levels are high, they oxidize hemoglobin inside red blood cells. So this forms clumps called Antibodies. These clumps weaken the red blood cell membrane, making it fragile. Eventually, the cells break apart in a process called hemolysis, where red blood cells are destroyed faster than they can be replaced. This leads to symptoms like anemia, jaundice, dark-colored urine, and fatigue. G6PD deficiency is often detected in newborn screening. With proper management, mainly avoiding known triggers, people with G6PD can live healthy lives. Throughout our biochemistry journey, we've seen how molecules keep you alive and how changes at the molecular level can impact your health. We started with carbohydrates, your body's main energy fuel and cell signaling components. We moved into lipids, essential for building membranes and storing energy. We explored around the world of proteins, from antibodies and storing energy. We explored the world of proteins, from antibodies and hemoglobin to enzymes that run your metabolism. And finally, we looked at what happens when those enzymes don't work properly due to genetics, leading to conditions like PKU and G6PD deficiency. All these concepts connect back to one big idea. Your health is written at the molecular level, and understanding that gives you power. And that's the end of our biochemistry enrichment class.